Okay. Yes. Okay. A internet tá aqui.
É sério isso?
Estranho.
A comida tá chegando. Parou o vídeo?
Is that these cells are actually shrinking. They're losing water. And what we discovered was that this change in the physical shape of the cells was actually also mediated by ATP. If you apply ATP to the preparation, all of these cells will shrink massively. If you block ATP receptors, they won't undergo this activity. And these shrinkage events are on the same time scale as the currents that we record. So there's some link between the two. And what was really interesting was when we looked at where and when these events occur, we found that they were occurring right next to where the hair cells were found. And basically monitoring these optical properties, because these cells are very sensitive to ATP, it reveals when and where ATP was being released within the cochlea. And you can see that these events are happening very close to the hair cells. In fact, even the fingers that kind of interdigitate between the hair cells were sometimes shrinking. So we thought, well, if ATP is being released here, perhaps it would have the ability to diffuse over and bind to purinergic receptors in hair cells. Hair cells have been shown to express purinergic receptors. And so we thought, oh, well, actually, maybe there is a means by which this spontaneous activity in the supporting cells could impact the hair cells. And so what we did then was to record from the hair cells in these preparations, and indeed you can see similar types of spontaneous activity in the hair cells. And remarkably, this is very dependent on the close association between the supporting cells and the hair cells. And so we have two forms of spontaneous activity, and the spontaneous activity in the hair cells was also blocked by purinergic receptors. But of course, these could be two different forms of activity. And so what we did was we recorded simultaneously from the supporting cells and the hair cells, and also monitored the optical properties. And you can see in this trace that there's very close coordination, one-to-one correlation between the currents in the supporting cells shown in red, the currents in the hair cells, and also these crenation or shrinkage events that happen in the tissues. So really, it's all the same type of spontaneous activity, just being manifested in different ways in these different cell types. And finally, we'd like to know, does this activity actually activate the spiral ganglion neurons? Because it's going to play a role in development. You would want it to activate these first-order sensory neurons so it can then be conveyed to the CNS. And so we isolated the cochlea with the spiral ganglion attached and recorded in a cell-attached mode from these spiral ganglion neurons. And if you do this, you can see that for long periods of time, these cells are very quiet, but these periods of quiescence are interrupted by periods of intense firing. And what we did was we then monitored the shape change in the supporting cells that were located near the hair cell, which was presynaptic to the neuron that we were recording from. And indeed, each one of these burst events was associated with a crenation in the supporting cells near the hair cell that was presynaptic. And we know that this is an indication, obviously, of ATP release. And so it shows this very strong correlation. And furthermore, these burst firings of the spiral ganglion neurons was blocked by purinergic receptor antagonists, and you could excite the neurons by applying ATP. So this creates a sort of model by which hair cells could be activated. And I don't have time to tell you about all the experiments that we've done over the years, but just suffice it to say that it's turned out to be a lot more complicated than we could have ever envisioned. We originally thought that these supporting cells released ATP, and ATP just diffused and bound to receptors on the hair cells and activated them. But it turns out that it's actually a very indirect method of activation, whereby ATP acts on an autoreceptor on the supporting cells, induces release of calcium, it activates a calcium activated chloride channel, and actually the excitation of the hair cells is not through ATP directly, but rather through potassium release from these supporting cells. And the sequence of events is shown here on the right, eventually leading to burst firing of the spiral ganglion neurons. And I just want to point out one important thing here, and that is that this mechanism of generating spontaneous activity depends on the hair cells. So in a sense, it is truly taking the place of sound. The supporting cells are activating the hair cells directly, they release glutamate and then excite the spiral ganglion neurons as if they had been activated by sound. And you can see this in this calcium imaging movie where we're monitoring the activity of the spiral ganglion neurons. You'll see groups of these spiral ganglion neurons become activated. And if you block synaptic transmission between the hair cells and the spiral ganglion neurons, you see the entire preparation then goes quiet because it shows that it's very dependent on this glutamate release. This is the group data showing individual activity in different spiral ganglion neurons, and you can see that there's this high degree of correlation between the cells, because when ATP is released, a whole bunch of these supporting cells get activated, they release potassium, and then it excites a group of hair cells located nearby, and then you get groups of spiral ganglion neurons activated. And you can see that this activity is blocked by ND2X. Cells are still there and responsive because they can be depolarized with high potassium. So in thinking about this, it's very surprising in the sense that it seems like it's way more complicated than you would expect. And this brought to my mind this American cartoonist who Reuben Goldberg, who used to create these fantastical contraptions to solve very simple problems. And this is an example of one of the cartoons that he drew, which was what he called an automatic dishwasher. And so what he did is develop a system where he knew when the parents left to go to a movie after dinner that the cat would get lonely and start to cry, and that would scare the mouse. And then he has a pulley system set up such that would trip the sprinkler system in the house to then wash the dishes. And once the water starts to flow, then the turtle is stimulated to go search for the river, and that trips on the heating element to dry the dishes. And so this is some ways, I think is kind of shows an elaboration of the cells in the cochlea to use a very complicated system. And I like this example because it involves sound and mice and fluid, fluid release from the supporting cells. And so it seems like it is an overly elaborate system, because why wouldn't the system have evolved just to use ATP to stimulate the hair cells? Well, it turns out that the cells that are next to the hair cells in the cochlea in this epithelium are really epithelial-like cells, and similar epithelial cells use this mechanism to form tears in the tear ducts, to secrete fluid in the gut and the colon. And so you can imagine that these cells sort of have a toolbox to work with. And they are normally designed to do this, but now they're using this mechanism. Instead of sort of secreting fluid, they're using it to secrete essentially salty tears, which then changes the extracellular potassium around the hair cells to cause them to be polarized. So in essence, it is a very elegant system, and the cells used what they had available to them to achieve this purpose. So let me tell you about some of the work we're doing to identify the molecular components involved in this process. So one of the things that is absolutely critical is this autoreceptor. ATP acting on these cells directly. So the cells release ATP, and then it binds to receptors on those cells. And we know that this is a metabotropic receptor because of calcium imaging experiments that we've done. And so we went looking for potential candidates, and using DNA sequencing, RNA sequencing databases that were available, developed by David Corey's lab at Harvard, what we found was that there was one particular isoform of purinergic receptor which was highly enriched in non-supporting cells, and the expression of this receptor went up during early development. And so that's extremely fortunate. It seems like there's one primary candidate. And so we obtained knock-in mice which express LAC-Z under the P2RY1 promoter. And then we immunostained the cochleas, and you can see the hair cells here. And in this Kohl's organ, this region of this tissue, the supporting cells that are adjacent to the hair cells, you can see that they have very strong beta-galactosidase immunoreactivity, indicating that these cells have the activity for the promoter for this receptor. And then we took advantage of an antagonist, which was originally developed for blood clotting, which is highly selective for the P2RY1 receptor. And you can see application of this antagonist inhibits this crenation and the shrinkage of the supporting cells. If you record from the supporting cells and look at their spontaneous activity, it is also abolished by this antagonist. And furthermore, if you record from the hair cells, you can see that even though hair cells don't have P2RY1 receptors, because their activity is dependent on the activity of the supporting cells, their activity is also abolished by this antagonist. So this really points to an important role for P2RY1 in the supporting cells in controlling this activity. And we've subsequently done conditional knockouts of this receptor and so forth. In addition, I didn't have time to mention that we've also identified the calcium-activated chloride channel, which is necessary to induce the chloride efflux. When there's chloride efflux, water goes with it, and that's why the cells shrink. And when you have anions leaving the cell, it also draws potassium out of the cell, and that's what causes the local depolarization. And so we've identified this particular channel, it's TNEM16A, and animals that lack this channel do not have the spontaneous activity in the supporting cells or hair cells. So all of the work that I've described for you up to this point was done in isolated cochlea. And you know, it is a very artificial system, right? We have to dissect it out of the inner ear. It's encased in bone, so it undergoes trauma. And it's, you know, it is a mechanically sensitive organ, so we can't avoid the possibility that we've induced some trauma. So we've been spending a lot of time in the last few years to take this work that we've done in vitro to in vivo context. And one of the ways we've done that is to use calcium imaging in awake animals prior to the onset of hearing. And we do this in this particular animal, which expresses a genetically encoded calcium indicator, GCAM6S, under control of a ubiquitous neuronal promoter, SNAP25. This is an animal that was generated by the Allen Institute. We then, you know, under anesthesia, we create a cranial window over the midbrain, which contains two sensory processing centers, the inferior colliculus and the superior colliculus. The inferior colliculus gets input from the auditory system, and the superior colliculus gets input from the visual system. And then we make time-lapse recordings from these animals in a macroscopic way. And in these animals, it's quite remarkable that you can see correlated activity of neurons in animals that are blind and deaf, but you see this activity in the visual system and the auditory system. So it's as if they're experiencing sound and vision, even though they're not doing this at this time. And if you look at these regions, you can see very different discrete forms of activity. The activity in the superior colliculus propagates as waves, and that's actually a reflection of the retinal waves, the waves of activity that's passing through the retinal ganglion cells. And if you look down here in the inferior colliculus, you can see it's very different looking activity. It's sort of bands of activity. And I can illustrate that a little bit more clearly here in this movie, where we're just now looking at the inferior colliculi. And you can see that there's these sort of chopper-like activity patterns. And remember, this is very macroscopic. We're looking at the entire midbrain here. So we're not visualizing individual neurons here. It's actually the clusters of neurons and afferents that are active at the same time. But it shows the very discrete nature of this activity. It's spontaneous, it's balanced, it sort of occurs on both left and right side. And if we do 2-photon imaging in vivo, we can see that this activity corresponds to increases in calcium within neurons. And these are, very discrete bands of activity such that neurons that are almost adjacent, to other cells show discrete forms of activity. You can see that here in these 3 different colors. So these are 3 discrete events, and you can see that the neurons in these regions exhibit correlated activity along the band, but not correlated with cells nearby. And so why this is interesting is because the auditory system is organized in a tonotopic way, such that cells that are responsible for processing certain frequencies of sound are clustered together. And the tonotopic axis goes in this direction. So you can imagine that frequency varies over the surface of the colliculus. And so what this is illustrating is that basically these cells are experiencing tones of sound, essentially. So you can imagine that that these are neurons that will ultimately process similar frequencies of sound, and they're being induced to fire in a coordinated way during this period of development. We all know about Hebb's correlates about cells that fire together, wire together, and so we have a mechanism here by which these neurons that are going to be important for processing similar frequencies of sound are showing correlated activity. And you can see that some discrete events here. Sometimes the bands are in the middle of the inferior colliculus. Sometimes they actually bifurcate and form doublets. And even though each of the events is discrete, over time, you basically have all of the neurons within this region exhibiting this sort of spontaneous activity. And I mentioned that this is similar to the tonotopic axis, and you can actually demonstrate this experimentally if you just wait a little bit longer for the middle ear to open up, and then the animals become sensitive to sound, and then we can play low frequency and higher frequency sounds to the left ear, and you can see because of the contralateral movement of information through the nervous system, that this stimulates intense activity in the middle region, so this would be the lower frequency encoding region. And then if you apply higher frequencies of sound, it bifurcates and you get the lateral activation. And the remarkable correspondence between the sound evoked responses and the spontaneous activity really illustrates what this intrinsically generated activity is trying to do. And so this leads us to the conclusion that these localized events within the developing cochlea are then propagated through the developing central nervous system up into the brain to these auditory processing centers to induce spontaneous firing of neurons that will ultimately process similar frequencies of sound. So if that's true, we should be able to do some relatively simple experiments to prove that the activity that we see in vivo is really generated by these mechanisms that I've described for you. And one of the ways that we look at this activity is to put regions of interest around the 2 lobes of the IC, and then integrate the activity over time. So this shows the fluorescence over time. And then we can represent individual responses by these horizontal lines, where the length of the line represents the magnitude of the fluorescence change. And because they're not absolutely mirrored with one another, often one side is larger than the other, typically because the input comes from one ear or the other. We give a dot here, and the size of the dot is an indication of the degree to which one side was larger than the next. So that's all just a means of kind of illustrating, I think, en masse, the nature of this spontaneous activity. And you can see that it's incredibly kind of stochastic and random. The events are all different amplitudes, and they, there's left and right bias. But if you integrate this over time, you can see that they're incredibly balanced, such that, all of the neurons in the left inferior colliculus and all the neurons in the right inferior colliculus are experiencing overall the same amount of activity, and the same amount of activity along this frequency domain. Now, if this input is really coming from the cochlea, if we surgically remove the cochlea, we should abolish the activity on the contralateral side. And you can see we remove the right cochlea, it really dramatically reduces the activity on the left side. And same, if we remove the left cochlea, it reduces the activity on the right side. And if we use a bilateral surgery, you basically abolish all spontaneous activity, indicating that truly this activity does have its origin within the developing cochlea. It's not something that's intrinsically generated within the brain, for example. So like the retina, this spontaneous activity in the auditory system comes from the developing peripheral sensory organ. So can we tie this into the supporting cells and the mechanisms that I described to you? So one of the ways that we've done this is to use this MRS 2500 compound. The barrier between the circulation and the cochlea is really not that mature during this early period of development, and we're able to get drugs into the cochlea. And so we apply this MRS 2500 compound through IP, and we monitor activity in the colliculus and in the two colliculi. And I'll speed up this movie here because we don't want to watch it in slow motion. But here's all the spontaneous activity in the IC. Here's the superior colliculus. Remember, this is the visual input. And what you'll see over time is that after we give an injection of the MRS 2500, the activity within the inferior colliculus selectively is depressed. Of course, we don't know how much MRS 2500 we're able to get into the developing cochlea, but it shows that this activity is very sensitive to antagonists of this P2R1 receptor, and this doesn't appear to be an effect that's happening in the brain because the activity in the superior colliculus is unaffected by this manipulation. And sham controls don't have any effect. We can record the activity over long periods in vivo, and this is the group data showing the sham injection versus the MRS injection for the colliculus, looking at the frequency or the intensity of the events, and we compare that with the lack of effect that's happening in the superior colliculus. So this really ties the spontaneous activity now to the supporting cells, and it shows that these non-sensory cells are playing a very important role in controlling development. Now, one of the things that we can also do is a more dramatic experiment. We can actually block glutamate release from the hair cells by using mice that lack a vesicular glutamate transporter. So these animals are deaf. If you record auditory brainstem responses, they have none. If you actually record from these neurons, these postsynaptic neurons, they have no synaptic responses. So as you would expect, because the hair cells require BGLUT3 to load glutamate into synaptic vesicles, they're basically shooting blanks and they are not able to excite the neurons. So the prediction we would have is that, because I told you at the very beginning that the spontaneous activity requires the hair cells, right? It takes the place of sound. And because of that, we would expect that mutations that affect these cells, mutations in BGLUT3 and other things, would basically block the spontaneous activity. So we would predict that there would be no activity in these animals centrally. And so we generated these mice and then did the same types of recordings, and were very surprised, as you might imagine, to see that the spontaneous activity persisted. And not only did the activity persist, but it looked very similar to the activity that we would record in a control animal. You have activity in the inferior colliculus, it's transient, it's oriented the same way, it has left-right bias, all those kinds of things that are hallmarks of the normal spontaneous activity. But how can that be? So there are some changes. You can see the frequency is a little bit lower, and there's some other less dramatic changes, but overall the pattern looked very, very similar, which just does not make sense. How could they do this without having the hair cells? Well, one clue that we got was when we went back to recordings that we had done earlier, where we had recorded from these postsynaptic neurons, we found that they were depolarized by when ATP was released in the preparation. And it's not hard to understand why that occurs, because when you have one of these events in the supporting cells, it causes the release of potassium. And potassium will depolarize anything in the vicinity. So the postsynaptic neurons actually do get directly depolarized by the potassium. But that depolarization is normally not sufficient to bring those neurons to threshold. they need the synaptic input to cause them to depolarize. And so one of the things that we thought about was that perhaps under conditions where there's no glutamate release from the hair cells, that these postsynaptic neurons might increase their excitability and actually be directly depolarized by this potassium release. And so we've done experiments now to look at this. In the VGLUT3 animals, even though there's no glutamate release, we can see coordinated activity of the spiral ganglion neurons when we do calcium imaging in these preparations. And here's an illustration of that. So here are the fluorescence traces from 50 individual spiral ganglion neurons, and you can see that their activity can be coordinated. There's also these unusual plateau calcium increases that we see. And obviously, because there's no glutamate release, this activity is not sensitive to an antagonist of AMPA receptors. So unlike what I showed you earlier, where the activity was completely abolished by NDQX, here the activity is totally insensitive. So there's some means for activating the cells which is not dependent on that. And we propose that it might be potassium. So you can see the number of events, the number of cells per event is lower. It's not as strong as excitation as you would see. It doesn't propagate over as large an area, and there's this appearance of these very long duration calcium transients. And so one of the experiments that we did to tie this back to the supporting cells was to use an antagonist of this calcium activated chloride channel, which we know is necessary to generate the potassium transients. So if potassium transients are directly depolarizing these cells and causing the excitation we see here, we block this channel, it should abolish the activity of the spiral ganglion neurons. So we use this compound, BBE, and it is a selective antagonist of these channels. And you can see remarkably it abolishes this activity. And the neurons are still there because if we depolarize them with high potassium, you can see a large calcium increase. So this illustrates that, under these conditions where there's no glutamate release from the hair cells, the spiral ganglion neurons can still be excited. So the system has its own mechanism to make it very, very robust and to preserve the spontaneous activity. Now, how do they do this? Well, one of the ways that they do this is by regulating the excitability of the spiral ganglion neurons themselves. And I alluded to this. And so we looked at this directly by recording whole cell from these spiral ganglion neurons. So normally in a control animal, the spiral ganglion neurons are not that excitable. Actually, it takes about 80 picoamps to generate a single action potential from these cells when you depolarize it. And they have input resistances that are kind of intermediate. If you look at it at peak or steady state, maybe 500 megohms, something like that. Now, if you look at these neurons in the knockouts for VGLUT3, right away you can see that they're much more excitable. To a much smaller current injection, you have many action potentials that are being generated. And if you look at the input resistance of the cells, it is increased dramatically. So at steady state, you have about 1.5 gigohm now. So even a small amount of current is able to have a much more effective depolarization of the cells. So they become more sensitive to that potassium that's being released by the supporting cells. And this is just a lot of group data showing the threshold where action potential generation has decreased in these supporting cells. So they get excited more readily. They have higher input resistance. Their membrane potential, so resting membrane potential, has not changed. But we also see this plateau behavior in the cells, such that if we inject current, sometimes they will depolarize, fire a bunch of spikes, and just stay at a depolarized level. And we can see that induced, and they also exhibit spontaneously. So very similar to what we see when we do the calcium imaging from these cells. So I want to kind of end here by telling you what we think that this activity is doing. Now, our early experiments on this, which I'll describe, were focused primarily on events that are happening within the developing cochlea itself, within the developing sensory organ. And we've used, in collaboration with Decibel Therapeutics and my colleague Uli Mueller, single-cell RNA sequencing approaches to look at the specification and maturation of these spiral ganglion neurons. And so this is isolation of cells in the developing cochlea, and you can see that there are neurons in here. There are type 1 and type 2 SGNs. I didn't describe that there are two different main classes of spiral ganglion neurons, but the only ones that were really important here for encoding sound are the type 1 spiral ganglion neurons. These are the ones that innervate the inner hair cells. And furthermore, you can subdivide these into 3 discrete types, 1A, B, and C, and that is based on different patterns of gene expression. And in particular, one can identify these different subtypes based on sort of key marker genes that are expressed by these cells. So the reason I go into that is because you can now use immunostaining to identify these discrete types of spiral ganglion neurons. And what we know by doing immunostaining at different times during development is that initially these spiral ganglion neurons are not well specified. And they actually start to exhibit this maturation specification over this early period of development. And the important thing I want to point out here is shaded in light blue, and it shows that a lot of the specification, you can see the percentage of cells that are expressing these different markers, actually happens prior to the onset of hearing. So mice begin to hear at about P12, something like that. And so you can see that there's already these dramatic changes in the expression of these different marker genes which identify the discrete types of spiral ganglion neurons. And much of the specification is actually happening before the animals actually experience external sound, pointing for a role of the spontaneous activity in inducing this maturation. And so we looked at the patterns of activity that are exhibited by the spiral ganglion neurons in these animals. And as you can see, the VGLU3 knockouts, the cells are much more active because they're more excitable, and the bursts take on a different structure from what is seen normally. And if we then look at the specification of the cells by using the calretinin or CAL2, calbindin-1, CAL4F1, and NGFR, you can see that there are significant changes in the expression of these key marker genes, suggesting that the specification of the spiral ganglion neurons has been altered when the patterns of activity that these cells are exhibiting has been altered. And so this points to an important role of this burst firing in this very discrete stereotyped manner in driving the maturation of these cells. And we've now gone to look at a number of other mutations that affect different aspects of the signal transduction apparatus and things like that. And we find similarly that it affects the specification, but does it in unique ways. Again, pointing to the idea that it's really the pattern of activity that matters. And finally, I want to just sort of project into the future and tell you, because we are very interested in neuron-glial interactions, we're obviously quite excited about the idea that this neuron-glial interaction may not be just restricted to the developing sensory organ, but actually may also occur within the CNS. And of course, I'm sure all of you know that astrocytes are the primary supporting cell type in the nervous system, and they ensheath synapses. And interestingly, astrocytes and neurons develop in parallel, and astrocytes have been shown to secrete factors that are really important for forming nascent synapses. And they've even been shown to engulf synapses and initiate remodeling of these nascent circuits. And so we wondered whether or not there was coordinated activity. So we've generated mice that now have genetically encoded calcium indicators expressed only within the astrocytes. And what we find is that the astrocytes also exhibit this highly spatially restricted spontaneous activity. And this activity is much lower in amplitude than what you see in neurons, which is why you see a little bit more background activity in this movie. But if you look at the patterns of it, it is very characteristic and very similar to what we see in the neurons, that it's spatially restricted, and it's also aligned tonotopically to the neuronal activity. And now we've done simultaneous recording from the neurons and the astrocytes in the same animals using 2 different calcium indicators. And you can see that the astrocytes and neurons in the same domains show correlated firing. The astrocyte activity is lower frequency because it has a higher threshold, and it's longer in duration than the responses in the neurons. And finally, what we've, I can skip that and go to the kind of the crux of it, and that is we've identified the mechanism of this. So it involves the activation of metabotropic glutamate receptors on the astrocytes. So when neurons show correlated firing, a lot of glutamate is able to diffuse out of the synaptic cleft, and it can reach the membranes of astrocytes, where it can bind to metabotropic receptors, in this case mGluR5, and then induce a calcium rise within the astrocytes. And we can antagonize this mGluR5 receptor. It completely abolishes the responses in astrocytes. And if we do selective genetic deletion of mGluR5 from astrocytes, it also completely abolishes the responses in astrocytes, showing that astrocytes and neurons are showing highly correlated activity during this critical period of development. And we think that that may be important for coordinating the activity of astrocytes at the same time as neurons to induce synapse formation, synapse refinement, and so forth. So let me just summarize some of the main points of the things that I've told you about today. So one is that spontaneous activity in the auditory system, it occurs through a very unusual mechanism, and it involves the release of ATP from these supporting cells that are located immediately adjacent to the hair cells. So it's not the hair cells themselves, they take the place of sound, and they do this by releasing ATP. I showed you how spontaneous activity is able to persist in animals that carry deafness mutations. In this case, we looked at a VGLU3 knockout mouse, but we've looked at a number of other knockout mice from mice that they will be deaf after the onset of hearing, but during this developmental period, they exhibit robust spontaneous activity. And that is, I think, very important because we know that many patients who carry these mutations, the children are good candidates for cochlear implants. So this is a prosthetic device that is placed inside the cochlea, which directly stimulates the spiral ganglion neurons. And we think that preservation of this spontaneous activity may make these children good candidates for cochlear implants because they've been able to experience this early period of activity, which is important for neuronal survival and specification and maturation, as I showed you. I showed you how spontaneous activity promotes maturation and specification of the spiral ganglion neurons. And finally, I showed you how neurons and astrocytes are exhibiting coordinated activity. And this is some of the work that we're really focusing on now, is to understand how this activation of astrocytes stimulates their own maturation and perhaps allows astrocytes to promote development of those circuits. And finally, I just want to project a little bit more into the clinical realm and mention that most of the inherited forms of deafness involve mutations in connexin 26. And connexin 26 is a gap junction protein. And why this is interesting is because connexin 26 is exclusively expressed by the supporting cells. It's not expressed by hair cells, it's not expressed by the spiral ganglion neurons, it's just within the supporting cells, and it's a leading cause of deafness. And we don't know how it is that mutations in this lead to deafness. But one of the things that, as far as the mechanism is concerned, we believe that connexin 26 hemichannels may contribute to the release of ATP from the supporting cells. So potentially that could contribute to the impaired maturation of the cochlea in these animals that carry these mutations. And finally, I wanted to mention tinnitus. Tinnitus is the perception of sound where there is no sound. Most of us have experienced this as a ringing in our ears. It happens after you've been exposed to a loud sound or gone to a concert, indoors, that kind of thing. It's loud. You have this phantom sound perception, and it can be extremely debilitating. And what I've described for you is a mechanism by which sound activity can be generated in the CNS in the absence of sound. So basically, it's an intrinsic mechanism within the cochlea to produce the perception of sound. And although we don't know that this contributes to tinnitus, certainly changes in the cochlea are important for initiating tinnitus. And we think that it's possible that injury and trauma to the cochlea could actually reinitiate some of these developmental programs to reinitiate release and so forth to contribute to the direct activation of the hair cells and the spiral ganglion neurons. So that's something that we're hoping to continue in the future. So to conclude, I just want to acknowledge the extraordinary individuals who have contributed to this work in my lab, a number of graduate students and postdocs. In particular, Travis Babola was responsible for all of the P2RL1 data that I showed you. Han-Chin Wang, who's now a postdoc at Berkeley, was responsible for identifying TMEM16As, the calcium activated chloride channel, and the potassium release phenomenon. Nick Tritch was the first student who really made the discovery about the crenation events in the cochlea and launched us on this exciting research direction. And Verick Fellner has been just a postdoc in my lab who's been doing the work on astrocytes. Very grateful to our many collaborators over the years. And with that, I'll finish, and I would be happy to take any questions.
Thank you. Amazing talk. Let me get the chat here, and also there's several questions here.
Sure.
I'll leave mine to the end then. Ricardo Reis, do you want to ask your question, or should I?
No, I can ask. Many thanks for your talk. Amazing talk. I really enjoyed how such complex this circuit that you could pinpoint the major elements. And one question is, if I understood, the first part of your talk was more developmental related. And I would like to know if you ever have done experiments where the sounds of nature could be given to the animals to see how the phonotopic cells are arranged. And if you can repeat that on animals that lose the capacity of hearing, are deaf in the adult, and you could somehow revisit and recapture this kind of sensorial element in the adult animal that is deaf.
That's a great question. And so I think that the really core of your question is, can we understand what the consequences would be if you did not have this early spontaneous activity? Or if this early spontaneous activity was in some way fundamentally altered by having a mutation or whatever. And so one of the main challenges has been that all previous studies in this kind of area have involved removal of the cochlea or analysis of mice that are deaf. And so if the mice are deaf and they have no cochlea, you cannot measure their hearing. You cannot play normal sound, you can't do anything. So you're exactly right, and what we have been doing is really to try to drill down into the mechanism of the cochlea so that we can do selective manipulations of the spontaneous activity, yet allow the animals to hear later. And then so that we can do things like assess their sensitivity to sound, assess their ability to discriminate similar frequencies or interpret complex sounds like the communication that they give to each other, those kinds of things. And so we have embarked on those studies now. So in particular, we're using conditional knockout of the TMEM16A channel. So we delete it specifically from the supporting cells. You don't have the spontaneous activity. And now we've been looking in vivo at the response of those animals to sound. And I can tell you that there's lots of changes. I mean, even though the animals, the eighth nerve is still there, the cochlea is still there, the neurons are still there, the wiring of the circuit in the auditory system is fundamentally altered. So we can see changes in the sensitivity. So there's, one of the things that we think this spontaneous activity do is to sort of create the proper homeostatic scaling of the strength of synapses in the auditory system. And so we see alterations in that. We also see alterations in the sort of inhibitory circuits that are found in the CNS. So that's work that's ongoing, but I think that's really the critical question that we would like to understand is, what aspects of development is this spontaneous activity involved in, and that's the approach that we're taking. And in fact, that was really a lot of the motivation for doing the hard work of uncovering all of the detailed mechanisms that I described for you. That's once we understand that, we can then make sort of surgical manipulations in the cochlea to ask what the role of activity is and how it is that these bursts actually initiate development.
Many thanks. Amazing.
Thank you. Claudia Vargas, do you want to put your question? I don't know if you're there. She asked if spontaneous activity found in the inferior colliculus after peripheral lesion scales with the rat age.
It's a great question too, and that is, yes, so the activity does change during development, and it's not constant, and one of the reasons for that is because the cochlea itself matures. And it matures at a different rate in a base to apex. So you have the coil, and the base is responsible for encoding high frequency sound, the apex low frequency sound, and during development there's a developmental gradient where the base is developmentally older than the apex. And because the activity sort of turns on and then evolves, what you see are different patterns of activity in the high frequency regions versus the low frequency regions, and it evolves over time in the animals. So yes, it does sort of ramp up, and I don't know if I have a slide. Sometimes I get this question, and that is related to that, and that is, well, how does the spontaneous activity go away? Does it ever go away? And that was actually one of the remarkable things. If you now record from the cochlea from a hearing animal, so just after hearing onset, let's say P14 or something, you record from the supporting cells, the hair cells, or you look for these changes in shapes of the remaining cells that are adjacent to the hair cells, all of these features of the spontaneous activity are gone. And that makes sense, right? You wouldn't want spontaneous activity competing with sound-evoked activity. Now, it's not a perfect system. In those early ages, right after the opening of the ear canal, we can see sound-evoked activity and spontaneous activity coexisting for a day or two. And then, basically, you only get sound activity, and the spontaneous activity is shown here. It just disappears within the cochlea. We think that the main reason for that is that the spontaneous release of ATP is what goes away. Because we can, in these preparations where we don't see any activity, if we apply ATP, we can show that the cells still have the receptors. They're still responsive during this period. So it appears that that initial release process is what goes away first, and that's what silences the intrinsic activity.
So is connexin 26 still there?
So connexin 26 comes on and it's expressed throughout life, yeah. But so I'm speculating here, and we don't have conclusive evidence for this, but a lot of circumstantial evidence that it is likely that ATP is coming out through a channel, and we think that because if we block vesicular release or vesicular loading of neurotransmitters and things in the preparation, we can block the spontaneous activity from the hair cells, the spiral ganglia neurons, so we know it's working, but it does not block the spontaneous activity in the supporting cells. So there's still ATP release, doesn't seem to be sensitive to things that block secretion, which makes us think that ATP might be coming out through a channel. And we know that gap junctions can exist as hemichannels. You might have heard of that. So basically, gap junctions work by pairing between cells to create an intercellular channel. But occasionally you can have unpaired connexins, which create a large pore at the surface. And indeed, if we had one of these channels that was open just for a fraction of a millisecond, it would allow the ATP release, which could induce every one of these things. And these supporting cells are expressing enormous amounts of connexins during this period of development, and they're inserting in the membrane, and then they're linking up to their pairs. And so it's possible during this period of development that you have more of these unpaired connexins that just through probabilities, they just occasionally open, and that is enough to allow the ATP to come out, and then eventually these things are all linked up to their mate, and they no longer have the hemichannels present at the surface. So So this is a leading hypothesis that we have right now that might actually be the generation of these large connexin plaques that is part of this mechanism, which is, again, it's kind of an amazing thing. It already has this, developmental sequence where they have to establish this large syncytium, and so it's just using these connexins maybe to initiate the ATP release. But there's clearly a lot more work that needs to be done there. We've looked at connexin 26 knockouts, and the activity is different. It's suppressed, but they still have some spontaneous activity. But there are other connexins that are also expressed during this time, connexin 43 and connexin 30. So many different sort of channels could be candidates for ATP release.
Thank you. Now Maria Florentina is asking, if you abolish spontaneous activity in the cochlea during early development, is the topological organization of the auditory cortex disrupted? Also, is the frequency of spontaneous activity constant, or does it change according to different phases of development?
Okay, good. So I think I might have answered part of that, where it does seem to change over development. One of the things that people have often asked is, like, is there some information in the activity encoded in the frequency? Like, so, how does it, if it is important for like tonotopic refinement, then maybe you would need to have the high frequency area cells exhibiting different patterns of activity than low frequency cells. And as I described, that sort of happens because of this base to apex developmental gradient, which means that at any one time in development, cells in different regions are slightly in different states of maturity, and they exhibit slightly different patterns of activity. But prior to our work in this field, people had looked at deaf mice, and they had removed the cochlea from animals. And what you find is that there is still a tonotopic organization of the projections within auditory centers. So that's important. What it means is that this spontaneous activity is not like essential for creating the map. What we know about pathfinding and that kind of connectivity suggests that a lot of this is governed by these sort of genetically pre-programmed events. So it's like BPH, heparin signaling, gradients of growth factors and so forth, which are important for setting up the initial pattern. And so we believe that tonotopic organization is established by these pre-programmed events. But then you then layer upon that activity, and then you start to refine these a little bit, and you correct errors and so forth, and increase survival of neurons during this period where we're going to get a surplus of neurons. Many of them undergo apoptosis. So the question is, are there changes in animals where we disrupt this spontaneous activity? We can see the differences in the width of the bands, but it's still early days yet. I would say that that kind of analysis we're still doing in the mice, where we've done the targeted manipulations of the TMEM channel. We couldn't use the VQ3 animals because they're deaf, so we can't look at tonotopic organization of those animals. It's really required this conditional knockout approach, and it's just taking us a bit longer than we had hoped because of all the same things that I'm sure you're experiencing with COVID restrictions and stuff like that. But this is the kind of work that we're doing right now to explore these questions.
So how does this correlate with the language acquisition? I remember the study with Chinese kids that they can hear the R's really well, and then suddenly they lose that ability to hear the R's, because they actually change it for the L's, right?
Yeah, very interesting. I hadn't heard about that. So those kind of higher order things, I would imagine, are not based in the detection, right? They can certainly hear these things, and it's encoded, but then how that's interpreted deals with how sound information is processed in the cortex, for example. And there's just a lot of incredible complexity there. one of the things that we've seen, in these animals, and I illustrated that in the movies, is that there's this bilateral representation. So even though we know that each spontaneous event occurs very discreetly in one ear or the other, they're not coordinated with each other, it's a spontaneous event, we can see that each of these events is represented on both sides of the brain. And that's very different from the visual system. If you look at the spontaneous waves that are occurring in the retina, they're absolutely unilateral. They're on the contralateral side, you don't see a mirror image. So even at these very early stages, we see bilateral representation of basically sound information. And actually this bilateral representation we see is one of the most sensitive things that is disrupted when we put in mutations for various components of the transduction apparatus, the hair cells, or modulatory pathways. It seems to be very, very labile. So of course I have no idea in this specific example why they're not able to interpret ours, but I think it illustrates just the complexity of the sound processing system and the coordination that must occur across the brain in order to do this. And so, yeah, very, very interesting.
Thank you. Eduardo Sequeira wants to go for his question. Where is he?
Hi.
There he
Thank
is.
you very much for your talk.
Sure.
It was fantastic. So I'm thinking about the increasing activity of the sensory neuron in the Bergles knockout.
Yes.
And I was wondering if like, because there's this thing that during synaptogenesis, it was shown for some neurons that the postsynaptic neuron expresses many different neurotransmitter receptors, right? And then after the presynaptic neuron makes the contact, it restricts to the right receptor, right? Neurotransmitter matching. So I was wondering if in the absence of glutamate, maybe the sensory neuron keeps like receptors for ATP, for instance. And then there is a connection directly from the supporting cell to, activating the sensory neuron.
Yeah, that's a really good question. So could it be also that those spiral ganglion neurons increase their repertoire or haven't refined their repertoire of receptors, and could ATP be acting directly on those cells? So the only experiment that we did that would argue against that is when we used the BBE compound, which blocks TMEM16A, that abolishes the activity in the hair cells. And so that's downstream from the ATP release. So if ATP was acting directly on the spiral ganglion neurons, it wouldn't be sensitive to that. Of course, it's possible that there's ATP effects and the effects of potassium on the cells, and they work together. That's certainly possible. And it's also possible, that we're looking primarily here at calcium and using electrodes to record voltage. But of course, there's lots of signaling through metabotropic receptors that doesn't release calcium in the cells. It could be, changing cyclic AMP levels, which could be important for developmental maturation. So yeah, I think there's a lot of potential richness in other forms of signaling. Purinergic receptors, it turns out, there's a lot of them in the developing cochlea. I was discussing primarily this metabotropic receptor, but then there's a whole class of ionotropic P2X receptors, which are expressed on the hair cells and the spiral ganglion neurons, and even on the supporting cells themselves. And we have no idea what those receptors are doing yet, because when we block the metabotropic receptor, all the spontaneous activity goes away, even though there are all of these other receptors. So we don't know the conditions that are necessary for engagement of those other classes of receptors, and it's possible that in vivo context, those receptors could also be engaged, but we just don't see it in the ex vivo preps that we use. Maybe they're responsible for that residual activity that we saw in vivo. But yeah, really great question. I think it's, how cells respond when they're deprived of their normal input is a very interesting question, and we think is core to a lot of pathologies. I mean, homeostasis of excitation is really what breaks down in epilepsy. I mean, you just have inappropriate changes in excitability of cells, and certainly is very important for maintaining this narrow window where you can have plasticity happening in cells to encode memories and so forth. So yeah, we see it even at this core developmental level that homeostasis is very important.
Thank you. We have 4 more questions. Is that okay?
Absolutely.
Okay. So Luana Martins, I don't know if she's there. She says, considering these results, can the supporting cells be a new approach for cochlear implants?
Wow, what a great idea. I mean, it's a, you know, you could think about this, you know, our methods, cochlear implants are incredible. I mean, it's the most useful prosthetic. I mean, it's absolutely an incredible success, and these children do really well. But it's essentially just a wire, you know, that's inserted into the cochlea. And experimentally, people have been looking at using things like channel rhodopsin and expressing it into spiral ganglion neurons. And so one could imagine that, you know, given if we could envision using these kinds of genetic or viral approaches to express optosensitive or chemosensitive agents within supporting cells or other components, we could really evolve the nature of that activity. Because cochlear implants are not perfect and they don't last forever, and you know, they have challenges and they're not always predictable. The one thing that would probably prevent that is that hair cells over time, you know, in patients that carry mutations in connexin 26 and so forth, they degenerate. So you lose the outer hair cells first and then the inner hair cells. So those primary sensory cells are just no longer there. And actually, we don't know that much about what the supporting cells are doing after those cells are gone. I mean, the supporting cells are clearly still there. Whether they could be used to stimulate the spiral ganglion neurons in this way, interesting idea. I hadn't thought about that.
Thank you. Leo Morita, do you want to make your question? I don't know if he's here. So he asks, have you observed aquaporin knockout mice? Is this spontaneous activity still persisting on them?
Great idea. We haven't looked at any of the aquaporin knockouts, and we do think that, I mean, aquaporins are highly expressed in the cochlea. Aquaporin 1, I believe, is highly expressed in the supporting cells. And we do think that aquaporins are involved in these shape changes because there's a lot of water movement, you know, that causes these cells to shrink and crenate, and just passive diffusion of water through the bilayer would never be able to achieve that kind of flow over this time period. So we really do believe that this is facilitated movement, likely involving aquaporins, but unfortunately we haven't had access to those animals. But I am very excited about pursuing that question. We just don't have the tools right now, and there aren't good acute blockers for the aquaporins, unfortunately.
So Leo is in Mike and Nedegard's lab, so maybe those mice. So here we have a question from YouTube that comes from Catarina Leon. She's a neuroscientist in tinnitus. So she says, fantastic talk. So are you there? I think she's in YouTube. Do you think the spontaneous activity seen in the inferior colliculus in the V-GLUT3 knockout mouse is similar in temporal spatial patterns of activity seen in the inferior colliculus in any tinnitus model?
Right, that's a really important question. So one of the things about tinnitus, it typically manifests in the high frequency range. You know, it's high pitched activity, and you could imagine that if you're thinking about this being caused by changes in homeostasis, that the circuits that are important for processing high frequency sounds just might be more predisposed to being altered by this. We haven't seen anything in these early developmental periods which would suggest that kind of thing. But I will say that one of the things that we know about tinnitus, which often is a controversy a bit in the field, is that there were classic experiments in people who had uncontrolled tinnitus, and what they did was to surgically cut the 8th nerve. So there was no connection now from the cochlea to the brain, and some of these patients still indicate that they experienced tinnitus, so the perception of high frequency sound, for example, suggesting that over time, even though it may be initiated by changes in the cochlea, that tinnitus effectively moves into the brain, and that there are mechanisms there to generate the spontaneous activity. And we have seen in mice where we've done surgical removal of the cochlea, we have seen spontaneous events in the colliculus, which are remarkably similar to the events that we know are generated spontaneously, driven by the cochlea. And so that's really exciting, and it suggests some local mechanism for inducing the activity, and we're quite excited about whether astrocytes, maybe could be the source of this. Maybe they become reactive and can trigger one of these bursts, which then cascades into activation of neurons in a tonotopic way, giving the perception of sound. And so that's the kind of thing that we're looking into now, to look at changes in astrocytes as a result of this. But so far, at least in these developmental studies, we don't see hyperactivity in the high frequency regions selectively.
So phantom tinnitus, right?
Yes.
So last question. So Mateus Silva says, excellent work. Beyond support cells, cochlea has also Schwann glial cells. Do these cells have a role in cochlear development, and could they also be a target to therapeutic approaches?
Yeah, so the Schwann cells, it's obviously the peripheral nervous system, and they myelinate the type 1 axons. We haven't looked at this. Gabriel Correlated at the University of Michigan has been looking a bit at these cells, and we know, and this crosses over to the other interest of our lab, which is myelination. We know that myelination is dependent on activity. So it's quite possible that the patterns of myelin, or the timing of myelination of these axons, could be altered when they experience abnormal patterns of activity. We haven't looked at this ourselves, but it's a really interesting question. And in addition to the normal Schwann cells that form myelin, there are also these Schwann cell-like cells which surround the cell bodies of the spiral ganglion neurons. The spiral ganglion neurons are kind of like the dorsal root ganglion cells, except that they don't have this process which extends over to an axon. The action potential actually goes right through the cell body. So the cell body of the spiral ganglion neuron actually gets encased by a supporting cell. And those cells have some unique properties and are very poorly understood, and would be an area that's really, I think, ripe for some investigation.
Thank you, Dwght.
Thank you so much, everybody, for your great questions and interest in this topic. I really enjoyed presenting to you and also hearing about your questions.
Thank you so much. I can send you the questions later. Everybody's here. Amazing work, fantastic presentation. And I can send you the questions that were, but it's also, recorded, so you can also go for the YouTube to refresh the questions. Thank you so much, Dwght. I'll catch you for another, your promises.
Okay, yes. I'd love to get back and talk about oligodendrocyte progenitors and myelination.
Great. That will be the next topic then. Thank you so much. Thank you.
Thank you. Have a nice day, everyone. Take care.
Bye-bye.
Thank you. Bye-bye.
Bye.